G'day guys, welcome. This is M7.1, which is illustrating the mechanism of semi-conservative replication through complementary base pairing. So this is what you have to be able to do, illustrate, so you might have to, in some cases in a question and exam, draw it, but most of the time you just have to be able to explain it or be able to answer a question with an understanding of how this mechanism is working. I'm going to divide this up into two parts. Part one is going to be easy, so what is our main thing we want to take away from semi-conservative replication. And part B, or part two, will be all about the finer detail. So, for those students wanting a little bit more detail, a little bit extra, you'll get that in this video as well. Okay, so let's get started. So the first thing that we have to understand is that we originally have a cell. I'm just going to draw it in there. And this is the nucleus of a cell, and it contains 46 chromosomes. Okay. While we're developing and throughout our whole lives, these cells are continually dividing, making new cells to replenish the ones that are either old or damaged all the time. So before that happens, we can't just simply divide our nucleus and our cell into two and not do anything with the DNA, because if we do that, you'll see quickly what happens. So if we divide our 46 chromosomes that we've got in our nucleus without doing anything else, we're going to get 23 chromosomes. And then if we divide again, without doing anything to our chromosomes, we're going to get nucleuses with 11.5 chromosomes, and that can't happen. We can't have that. We can't actually really have 23 chromosomes in a proper somatic cell, which is our normal body cells. We can't have 23 to function. So we need our 46. So if we go back up, we need some way of getting that 46 chromosomes to go into our two new cells, both having 46. Okay, and the way that we do that is through DNA replication. So I'm going to draw another nucleus here. Okay, much bigger this time, and I'm going to focus on just one molecule of DNA. So normally our DNA consists of our two strands, or always, and on those two strands we know we have bases. Okay, we know we have an adenine base, we have a thymine base, we have a cytosine base, and a guanine base. And on the second strand, the complementary strand, we have complementary bases. And we know that adenine always pairs up with thymine, and cytosine always pairs up with guanine, and so on. And that's going to happen all the way along. So what happens during DNA replication? Well, it's very simple. Okay, so instead of having just our normal DNA with our two strands, what happens is we open those two strands up. Okay, so I'm just going to move down. So we're going to open those two strands up. So we've still got our A, T, G, C, G, T, A, A, for example. And on our complementary strand, we're going to have our T, A, C, G, C, A, T, T. But now these two strands, as they open up, they provide the template for a new molecule of DNA. So new nucleotides come in, and they match up with their partner. So adenine with thymine, thymine with adenine, and so on. That's a C, A, T, T, and on the other strand, A, T, G, C, G, T, A, A. So now if we count our molecules of DNA, I'll just put a little backbone on both of these. So now we've got 1 and 2. So we've got our 2 original strands, and now we've got our 2 new strands. Okay, and these are strands that have been built from the 2 strands as template strands. Okay, 3 nucleotides in the nucleus being paired up with their complementary base and providing a whole new molecule. So now, if we were to divide them, okay, then this could go to one cell, and the second part, so this part here could go up to one cell, and this part here could go down to another cell, and we actually haven't lost any DNA material. We've replicated our DNA, and now we can share it between new cells. So that's part 1. Okay, so that's a very simple thing. The thing to take away is the process is we've got our DNA molecule, it has 2 strands, both those strands act as a template for 2 new strands, and that happens through complementary base pairing. If there's any questions, feel free to place them in the comments below. But that's basically the gist of the really easy part 1 of DNA replication. Okay, so that's all you need. Hang on there. If you want to know a little bit more, continue on with the video. Okay, congratulations, you passed the test of continuing to watch the video, which is excellent. Especially if your name's Nick. Well done, Nick. Okay. So the first thing we're going to look at is structure of DNA, just to remind ourselves. So a normal structure of DNA has a deoxyribose sugar, which looks something like this. It's got a H in this position, which separates it from RNA. It's got an OH group in this position, which is the 3rd prime position. So this is a pentose sugar, 5 carbon. We have a phosphate, and if it's a free nucleotide, we're actually going to have 3, which provides our energy, and this is our 5 prime position, our 5 prime end. And if you notice about that ribose sugar is the end up the top here containing our phosphate is attached to that 5 prime end, and this hydroxyl group is attached to the 3 prime end. So we can then determine the direction of that molecule. We can either say it's a position from 3 prime to 5 prime or 5 prime to 3 prime. It lets our audience know, or the reader know, exactly how we're referring to it. So we know, or we should know, that our DNA molecule runs in opposite directions. So I'm going to draw the first one, and I'm just going to pause it because it's going to take a while. Okay, so I've got my ribose sugars placed in, and hopefully you can see which is going to be the 3 prime end of each molecule and which is going to be the 5. I'm just going to draw in the phosphates now. So we're going to have a phosphate there, and a phosphate there. We're going to have a phosphate there, and a phosphate there, and a phosphate there. Link to the 5 prime, the 3 prime, the 5 prime, the 3 prime, the 5 prime, and nothing on the 3 prime except an OH group, and 5 prime, 3 prime, to the 5 prime, to the 3 prime, to the 5 prime, and with an OH on the end. So this particular molecule is 3 prime to 5 prime, and this particular molecule is 3 prime to 5 prime. Very straightforward. We're going to have our bases in between. Okay, our complementary bases, and now we have our DNA molecule, double-stranded, and it's free to form that beautiful double helix that we've seen. Okay, so why is this important to DNA replication? Well, one of the strands is a lovely strand, and that's our 3 prime to 5 prime strand. And the reason why that is because the other strand runs in the opposite direction. So the first stage of DNA replication is to unzip this DNA molecule, and we use a very special enzyme called helicase. Okay, so this is helicase, and I might even type that out for you. So that's helicase. Okay, so that's helicase right there. And following helicase is a enzyme called DNA polymerase. Now DNA polymerase can only travel, I'll do them in black, can only travel along a strand going from 3' to 5'. Well, not that it can't travel in the opposite direction, but it can only add free nucleotides to the strand that's going from 3' to 5'. So as it moves along the direction that's shown here, as it moves along the two strands, it's able to freely add nucleotides to the 3' to 5' strand, but it can't to the 5' to 3' strand, and that's due to the way the nucleotides are arranged with the phosphate group attached to the 5', and that's where it gets its energy to form that bond. So because of that, this particular molecule at the top, this leading strand is what we call it, will be able to build along without any issues at all. So DNA polymerase, okay, DNA polymerase is then able to just move along and add bases, complementary bases to that 3' to 5' strand effortlessly, absolutely effortlessly, which is excellent. The annoying strand is this 5' to 3' strand, because DNA polymerase can't add free nucleotides to it, because it can only add in the 5' to 3' direction of that new strand. And because this is already 5' to 3', we know we're going to need to make a strand going that way from 5' to 3'. So that's annoying because our DNA polymerase can't do it. So what has to happen is we need another molecule, okay, another enzyme, and this particular enzyme is called RNA primase. This is Apple spell check. Primase. Okay. And RNA primase basically has to, okay, so let's get rid of this before, let's get rid of it. So let's just get rid of that. So before DNA polymerase can build from 5' to 3' in the opposite direction, RNA primase has to add on, let's say, 1,000 bases, like so, and then that then gives DNA polymerase a chance to join on and then go backwards. So a DNA polymerase can then use that, join on, and then build our complementary sequence. Oops. Our complementary sequence going from 5' to 3'. Okay, so to run you back through those steps, okay, Helicase unzips the DNA molecule, forming this replication fork, and if you can have a look at what that is through my notes or anywhere on the internet. Helicase unzips the DNA molecule. We now have two template strands for our new nucleotides. We've got one going from 3' to 5'. That's the awesome one because DNA polymerase can just race along adding free nucleotides to that one. The opposite strand, which is running 5' to 3', we need an RNA primase to run down a few nucleotides so that the DNA polymerase can add nucleotides back, and we call these whole fragments that get opened up over time, Okazaki fragments. Okay, and after basically going along, opening up a space, RNA primase, DNA polymerase, filling in the blanks, filling in that Okazaki fragment, DNA polymerase and helicase will continue moving along in this direction, opening up a new space. That has to be filled, and that continues to go on until the molecule has been produced and replicated. But it's all, the whole result is to make a DNA molecule that is now doubled, so that when we divide, we don't lose any DNA material. I hope that clears things up. You can look at the video over and over, and if there's any particular questions, ask either in the comments below or in class. Make sure you subscribe. Catch ya!